Let's look at the structure of DNA. So DNA, as a reminder for you, stands for deoxyribonucleic acid. You can see the D is for deoxyribo, the N stands for nucleic, and the A stands for acid. So you have DNA. Now, that's important to remember because you need to always remember that when we're talking about DNA, we are talking about a molecule that is a nucleic acid. So I often talk about DNA as being the section with lots of N words. Now, not rude N words, just a lot of scientific words begin with an N. And the first N word we actually have here is nucleic acid. So that's an important distinguishing factor as I go ahead and introduce the other N words. So this is the typical molecule of DNA. It has this coiled shape that you can see nice and clearly here. It looks like a twisted ladder. So the technical term that we use to describe the shape is a double helix. Double meaning it is made up of two strands, and helix meaning it is a twisted ladder shape. So when we are dealing with the process of DNA replication, this ladder will unwind to form a structure that looks more like this. So you still have this backbone of the blue structures here, which we'll deal with in a sec. It's called the sugar phosphate backbone, and it is joined, each strand. This would be the one strand that travels down like this, and then I'll highlight the other one, and the other one travels up like that. So if I highlight that one strand, it would be the same as that strand on the right in the right-hand picture. If we look at the sugar phosphate backbone, it's important that in the name of DNA, we have deoxyribo. So that is actually giving you the name of the sugar. So the sugar's name is deoxyribose sugar. Then we have the sugar phosphate backbone. So that phosphate actually is that scientific compound which stands for PO4 -3. You don't need to know that, but it's just important that you know that that backbone, this whole part here is made up of alternating sugar molecule, phosphate molecule, sugar molecule, phosphate. The sugar in DNA is always deoxyribose. Please note there's an OSE, which means it is a sugar like glucose, maltose, but in the name for deoxyribo, there is no SE there. Then we have, going down the middle here, we have these structures that we call nitrogenous bases. That is our second N word that we're going to deal with, but I'll get to that in the next picture. So this is one strand on the left, this is another strand on the right. These strands are what we call complementary strands. And when we say they're complementary, it means that what we see is that when there is a C nitrogenous base, there is always a G on the other strand. When there is an A nitrogenous base, there is always a T on the other strand. So that is what we call complementary base pairing. The C stands for cytosine, which always bonds with guanine, and the A stands for adenine, which always bonds with thymine. So this is what we call our complementary base pairing, with C bonding with G, A with T. You must know the letters and the name for what that letter stands for. This is a more scientific way of looking at the DNA molecule. So we still have a sugar phosphate backbone going down each side. That's one strand, that's another strand. There you can see the sugar molecule, and there you can see a phosphate. So it's phosphate, sugar, all the way down on both sides. So again, that sugar is called deoxyribose sugar, and we just got to label the phosphate molecule. Now we have here something called a nucleotide. So what this is showing us is showing you there's one phosphate, one sugar, and one nitrogenous base. And that is a nucleotide, which is our next N word, actually, before we get to nitrogenous base. So this is deoxyribonucleic acid. So that's our first N word, nucleic acid. The building blocks of DNA are nucleotides. So those are what we call the monomers. Like the building blocks of a protein would be amino acids, the smaller pieces. The monomer is the part that exists on its own. So you have these three molecules, a phosphate, a sugar, and a nitrogenous base. That is called a nucleotide, which all join together to make up the whole structure of DNA. So the nucleotides are made up of a sugar portion, a phosphate portion, and a nitrogenous base. It's important to note that the nitrogenous base is attached onto the sugar portion. This base is not connected to the phosphate. It is connected directly onto the sugar. These are the four bases in the key here. We call them nitrogenous bases. And those nitrogenous bases are adenine, thymine, guanine, and cytosine. So because of complementary base pairing, we always have adenine bonding with thymine, cytosine bonding with guanine. And the way that I remember that is you have the two pointy letters, A bonding with T and C bonding with G. And both C and G are curvy letters or rounded letters. So again, the structure of DNA, it is a double helix structure. At the moment, you can't see the twist because we've unwound the helix. It is made up of two strands, a strand on the left, a strand on the right. Each strand has a sugar-phosphate backbone where it alternates sugar, phosphate, and every single sugar molecule has a nitrogenous base attached to it. The nitrogenous base can either be cytosine, guanine, adenine, or thymine, but they will always bond with adenine joining with thymine, cytosine with guanine, and they bond by weak hydrogen bonds. That's the name of the bond, and you must know that. Adenine always bonds with thymine with 2 hydrogen bonds. Cytosine bonds with guanine with 3 hydrogen bonds. And those bonds are meant to be broken.